Hello, everyone. Thank you very much for joining us today at our Lipid Maps webinar. I'm delighted today to be introducing Professor Jean Vance from the University of Alberta. My name is Professor Valerie O'Donnell. I'm based at Cardiff University in the UK. And just a few quick housekeeping things before we go to the seminar. The webinar is being recorded. It'll be uploaded onto Lipid Maps if you can't make it today. As usual, it'll be about 45 minutes, 10 minutes for questions at the end. If you'd like to ask questions, please do post them in the chat or in the Q&A session during the talk. And then at the end, what we'll do is unmute you and you can speak directly to Jean and ask your questions at the end. And I want to thank Merck for sponsoring today's webinar. Very grateful for their support. So I'm going to go now ahead and introduce Jean. I'm absolutely delighted today to introduce Professor Jean Vance from the University of Alberta. Jean is originally Scottish and she obtained her BSc in chemistry from Bedford College, University of London, and a PhD in biochemistry from University of Pittsburgh. She's a professor, as I said, at Alberta, where she was a founding member with her husband Dennis of an interdisciplinary research group. And a couple of, I think, really key things to mention about Jean is one is that Jean and Dennis published a Bible of lipid biochemistry, which I've grew up as a lipid biochemist using all the time, and I'm sure many of us on this webinar did. And this is the biochemistry of lipids, lipoproteins, and membranes. And I noticed that the 5th edition was published back in 2008 with a huge number of really famous people as authors, and it must be time for an update of that, Jean. I hope you got plans to update the book, because that would be fantastic. Anyway, so that's the first thing. Second thing is that Jean's research, I think that's the really important thing we're going to focus on today. Jean's research focuses on intracellular trafficking of lipids in mammalian cells. And about 30 years ago, she really pioneered this area, really looking at how lipids are moved between different organelles, particularly the ER membranes, into mitochondria. And posted a link in the chat to an editorial that was published in Nature last year, which talks about Jean's early seminal contributions that really started this whole field, where it led and where it is now, where there's a huge resurgence and interest in this. And really, this is all about the idea that organelles talk to each other inside cells and how lipids play a major role in this. So check out that link because the article is there. So anyway, Jean and her lab isolated from rat liver a specific ER membrane fraction, and this is many years ago, associated with the mitochondrial outer membranes. And these mitochondrial associated membranes, or as called now MAMs, they are the site of PS synthesis, where it's then imported into mitochondria. So Jean studies were the first demonstration of a functional role for membrane contact sites in intraorganelle lipid trafficking. This is a really important contribution to our understanding of how lipids are organized inside cells. And she really led the field at this time, generating and characterizing mice lacking PS synthases, as well as mice lacking PS decarboxylase, in which mitochondrial function is severely impaired. In other studies, she investigated the synthesis and transport of cholesterol and phospholipids within astrocytes and compartmentalized cultures of primary mouse neurons. And some of these studies revealed a key role for Niemann-Pick C1 protein and cyclodextrin in cholesterol export from lysosomes. And as a recognition of her research on membrane contact sites and lipid trafficking, Jean was awarded the EMBO International Wilhelm Bernhard Lifetime Achievement Award in 2018. So I'm really looking forward to hearing about the historical perspective of Jean's discoveries into mitochondrial and ER trafficking of lipids. And I think it's worth pointing out a kind of a current interest in this really needs to come to, we're all very preoccupied at the moment with COVID, Jean. And as you know, these viruses get into cells and they interact with the ER where they actually take their lipids from. So I'm sure there must be some really interesting things to know about how enveloped viruses behave and how they interfere with the trafficking into mitochondria of lipids. I wonder whether you've got some thoughts on this. Maybe we could pick this up in the question and answer session. Anyway, I'm going to hand over to Jean. I've talked enough. I'm really looking forward to hearing her talk.
Hello. First of all, I want to say thank you so much for inviting me to give this talk. It's a real honor. I've known about lipid maps for a long time, and it's a real pleasure to be giving a talk with them. And also, I would like to make sure that I thank Merck, because they have been a long time generous supporter of lipid research, and we all thank them for their contributions. So my talk today, the first part of my talk is going to really be a historical background on how the membrane contact field developed with the isolation of this ER mitochondrial membrane contact site. Okay. So I'm going to give a short introduction on mechanisms of interorganelle lipid transport. then isolation of this membrane fraction, MAM, and then some ideas on the physiological importance of this pathway, and finally some other implications about membrane contact sites for the future. So first of all, so this is a fundamental biological question, and I have to say that there really is not that much known about this field at the moment. That is, how are lipids transported between organelles. Really, there is not a lot of information on this. So we know that the majority of lipids in eukaryotic cells are made on ER membranes, and that mitochondria have a limited capacity for synthesizing lipids, so that they must import most of their lipids from the ER. Of course, there are exceptions to this, like cardiolipin and phosphatidylglycerol. So my pointer. Okay. So this is a model of potential mechanisms of intracellular lipid transport. This is originally from Dennis Volker. I modified it just slightly. So in this model, this just shows the various options for how lipids could possibly be transported between membranes. One mechanism, so there's a donor membrane and an acceptor membrane. One mechanism, it would be a diffusion mechanism where a lipid would desorb from the donor and be picked up by the acceptor. So this is not very likely for lipids because they're generally not very soluble in water, and this would be an energy barrier to overcome. A second potential mechanism, and this is what was thought to be the primary mechanism 30 years ago, which is that a soluble lipid transfer protein would pick up a lipid from the donor membrane, travel across the cytosol, and dump it in the acceptor membrane. So this was an attractive model. There are some very abundant lipid transfer proteins like the PC transfer protein, non-specific lipid transfer protein in the cytosol. But this process has to result in a net transfer of lipids, and there's debate about whether this would occur or not with these proteins. And also when these proteins were knocked out, generally it didn't make much difference to the membrane composition in the cell. A third potential mechanism is a vesicle-mediated transport, where a vesicle buds from the donor membrane, fuses with the acceptor membrane, thereby delivering its lipids. And this does occur to some extent, but not for everything. And in particular, I think this is not a really viable mechanism for lipid transfer to mitochondria, as there's not a lot of vesicle transport going on there. The final mechanism is mechanism D, which is a mechanism whereby the two membranes, donor and acceptor, come close to one another or perhaps fuse. And this is what I have been particularly interested in. So we know that phosphatidylserine is made in the endoplasmic reticulum, and it must, and it has to travel from the endoplasmic reticulum to the mitochondria to be decarboxylated by phosphatidylserine decarboxylase. So what is the mechanism of this transfer of PS from the ER to mitochondria? We know that the PS decarboxylase enzyme is restricted to mitochondrial inner membranes. So this sets about to some studies we did. I wish to confirm that the synthesis of PS did occur on microsomes endoplasmic reticulum. So I isolated from rat liver homogenate, I isolated mitochondria and microsomes using standard centrifugation methods. And then, of course, performed enzyme assay for PS synthase on both of these membrane fractions. And I was actually horrified by this result, because I expected all the activity to be in the microsomes, whereas in fact there was at least as much activity in the mitochondria as there was in the microsomes. So this did not make me happy. I almost threw the whole thing out and gave up. But around that time, Ben de Cruijff in the Netherlands had published a new method for purifying mitochondria on Perkal gradients. So I tried this method and isolated and performed the Perkal gradient centrifugation, and ended up with two bands in the tube, a lower band, which is the purified mitochondria, and the upper band, which was this garbage that was contaminating my mitochondrial preparation. So I then proceeded to measure the PS synthase activity in these fractions. So if you look in pure mitochondria, now the mitochondria don't have this activity, very low. Whereas microsomes, and particularly MAM, they have high activity for PS synthase. So indeed, PS synthase is made in some aspects of the ER, but seems to be particularly present in this fraction of MAM that is attached to the mitochondria. And to confirm that we'd done our subcellular fractionations correctly, I assayed 2 ER marker enzymes and showed that they were abundant in the microsomes in the MAM, whereas the mitochondrial enzyme cytochrome C oxidase was particularly present in the mitochondria. Okay, so we looked at some other enzymes, other lipid synthesizing enzymes in the MAM. And so I isolated 2 rough ER, 2 ER fractions, one enriched in rough ER, one smooth ER, and also the MAM, and measured the activity of these enzymes: acyl-CoA acyltransferase, diacylglycerol acyltransferase, HMG-CoA reductase, and PS synthase. And you will see that the activities of all of these enzymes are much higher in the MAM than they were in the other 2 ER fractions. So for those of you who aren't familiar with phosphatidylserine too much, there are 2 enzymes that make phosphatidylserine in mammalian cells. They exchange the head group of a pre-existing phospholipid for serine. So PS synthase 1 catalyzes synthesis of PS from PC, whereas PS synthase 2 catalyzes the exchange of serine for the head group of PE to make PS. And another enzyme that we were interested in, because Dennis's lab was next door, was the PE methyltransferase enzyme. And this converts PE to PC, and this activity is also high in the MAM. So from these experiments, we considered that the MAM was a specific ER domain that has high affinity for mitochondria and high lipid biosynthetic enzyme. So what's it doing? And we went back and remembered these wonderful, beautiful EM studies that had been done in the 1950s. For example, this one from 1955, an EM of liver. I think it was rat liver. And this is showing that there are regions where the ER does come very close, does sit very close to the mitochondrial membrane. And there are many of these studies. Most people thought these were just artifacts, of course. But it might be relevant. And in addition, there were 2 other studies, at least 2 other studies, showing that people had isolated ER fractions that seemed to be associated with mitochondria. So what more do we know about the MAM? Is there a marker protein for the MAM? So in this case, we're looking at the enzyme PE methyltransferase again. Dennis's lab next door had generated an antibody, a very nice antibody to this enzyme. Incidentally, this is a protein that is present really only in liver in large amounts. So we isolated many subcellular fractions from rat liver and immunoblotted with the antibody. So these are 2 ER fractions: plasma membrane, nucleus, Golgi, crude mitochondria, pure mitochondria, and MAM. So you will see that this protein seems to be a very good marker for the MAM and is only in the crude mitochondria, but not in the pure mitochondria. And incidentally, is not really abundant in the regular ER fractions. We also use this antibody in for immunofluorescence and electron microscopy studies. I can't see. Okay, so so this again shows, this is another immunoblot, shows that the PEMT is abundant in the MAM, not in the bulk of ER. And also we obtained an antibody against PS synthase 1 from Masahiro Nishijima in Japan. And it showed that this enzyme, this protein, is also highly abundant in the MAM compared to the bulk of ER. And in addition, PS synthase 2, the other PS synthesizing enzyme, is also enriched in the MAM compared to the rest of the ER. So in conclusion, for this part, we think that MAM is a distinct region of the ER that has mitochondrial affinity. And these two membranes are closely opposed, but they are not fused. And we know this because we can separate the MAM from the mitochondria. Later studies showed that large regions of the mitochondrial surface is very close to the ER, within 20 nanometers. And we know that PS biosynthesis is enriched in MAM. And we also analyzed briefly, in old-fashioned methods, we looked at the protein and phospholipid composition of the MAM compared to those of the ER and the mitochondria. The phospholipids, by the way, for you lipid people, we use TLC to determine the lipid composition. Does anybody know what TLC is these days? I don't know. That's how it was in those days. Anyway, we found that these compositions were similar to those of the ER, not the mitochondria. Now, the main question, do these membrane contact sites, MAM, have any role in cell biology? So, of course, when we first discovered this in 1990, there was a lot of skepticism that they had any importance, thought it was just an artifact, and they were ignored for decades, I think, this report. However, it is a common feature of all plants, mammals, and yeast to have these membrane contact sites. So we set out to determine if there was a role for MAM in the import of PS into mitochondria. So from many experiments using reconstituted organelles and permeabilized cells, it was determined that this transfer was not mediated by diffusion, just diffusion of the lipid, by soluble lipid transfer proteins, or by small vesicles. There was some other mechanism in play. And of course, we thought of these contact sites that could eliminate the energetically unfavorable transport of the lipids through the cytosol. So we set up an assay for measuring the import of PS into mitochondria. So we used radio labeling, which was very common in those days. And so we start by giving serine, radio-labeled serine to cells. This is converted by the PS synthases into PS, which we think takes place in the ER and MAM. The PS is then transported into mitochondria, where it's decarboxylated to phosphatidylethanolamin by PS decarboxylase. So the formation of radio-labeled PE, starting with serine, indicates that the transfer of PS to mitochondria must have occurred. And we also know from other studies that this is the slow step in this process. So this is a good assay for measuring the import of PS into mitochondria. So we started with the crude mitochondrial preparation that we originally isolated that contains the mitochondria and the MAM. And we added radio-labeled serine and looked at the conversion of that serine into phosphatidylserine. Happened quickly. As this declined, radio-label appeared in PE and then in PC. So this crude mitochondrial preparation has the capacity to synthesize all of these three lipids from serine. So this is a little more complicated experiment. So in this experiment, we wanted to show that the import of PS into mitochondria occurred through the MAM. So in this experiment, we pulse labeled CHO cells with radio-labeled serine for 1 hour. Then we removed the radio label and chased the radio label for 8 hours, 7 hours through, and isolated subcellular fractions, microsomes, MAM, and mitochondria at these times. And then we isolated radio-labeled PS from these fractions. So if you look in the control cells, the open circles, radio-labeled PS was in the microsomes, it was in the MAM, and it was imported into mitochondria over time. However, if we depleted ATP from the cells using potassium cyanide and potassium fluoride in the closed circles, you can see that the radio-labeled PS no longer was present in the mitochondria, but instead had accumulated in the MAM. So it seemed to be a blockage in the transport of the PS from the microsomes to the mitochondria. So this indicated to us that the MAM was involved in this process. We also showed that in the same experiment that the production of PE was reduced when ATP was depleted, and also when we depleted the activity, decreased the activity of PS decarboxylase by using hydroxylamine, we also obtained this accumulation of PS in the MAM. So another indication of the importance of the MAM and this biosynthetic pathway for mitochondria is that PE is enriched in mitochondria relative to other organelles. But what is the biosynthetic origin of this mitochondrial PE? There are two, as all of you will know, there are two main pathways for production of PE in mammalian cells. One is the CDP ethanolamine pathway, the last step of which occurs on the ER. The other pathway I've just talked about is the PS decarboxylase pathway, which occurs in mitochondria. So which of these two pathways provides the most PE for mitochondria? So to answer this question, we did an old-fashioned radio labeling experiment. We looked at the incorporation of ethanolamine as a marker of the CDP ethanolamine pathway into PE in the various fractions in CHO cells. We used a pulse chase experiment, pulsed for 1 hour with tritium ethanolamine and performed a 24-hour chase. We isolated mitochondria, microsomes, and MAM at these different time periods. And what we found was that the mitochondria, at the end of this time, even after 24 hours, there was almost no PE derived from the CDP ethanolamine pathway in the mitochondria. However, when we did the opposite experiment, where we radio-labeled the cells with tritium serine, you'll see very quickly serine-labeled PE appeared in the mitochondria. So this is the PS decarboxylase pathway that's making this PE. So from these experiments then, we considered that the PS synthesizing enzymes in the MAM made PS, which is imported into mitochondria. This PS is then decarboxylated by PS decarboxylase to PE, provides a lot of the mitochondrial PE. In contrast, PE is also made in the ER from the CDP ethanolamine pathway, but this source of PE does not readily become imported into mitochondria. And the same occurs in yeast. So, conclusion about this is we think that PS import into mitochondria does indeed occur via these MAM contact sites and requires ATP. Experiments I didn't show also revealed that newly made PS is preferentially channeled into mitochondria compared to old PS, pre-existing PS. And from some studies by Will Prince, we know that the import of PS into mitochondria is increased when the MAM are enriched in phosphatidylserine. And it's very similar mechanisms in yeast, as shown by many beautiful studies from the Volker lab and the Gunter Daum labs. So, is there any physiological importance to this pathway? So to address this question, we began by generating mice that lacked either PS synthase 1 or PS synthase 2. And actually, I have to admit that these were pretty disappointing experiments. These mice appeared rather normal. They bred normally. These mice were actually generated by Steve Young, and we collaborated in characterizing the mice. So in these mice, we'll analyze the phosphatidylserine content of their tissues. Seemed pretty normal. When we eliminated PS synthase 1, PS synthase 2 activity was increased and vice versa. So these two enzymes seem to compensate for one another. And when we crossed the PS synthase 1 and 2 knockout mice to try and obtain double knockout mice, we were unsuccessful. There were no double knockout mice. So this said that PS is absolutely crucial, or perhaps it's the PE that's generated from this PS. So we go next into the PE biosynthetic pathways. Maritza Bakovich's lab in Canada, she had made a global knockout of the cytodilyl transferase in the CDP ethanolamine pathway in mice. And this was embryonic lethal. So this pathway for PE synthesis is essential for viability. We made with Steve Young, we made the PS decarboxylase knockout mice. PISD, by the way, is the gene name for PS decarboxylase. These mice died early during embryogenesis. The heterozygous mice seemed particularly normal, almost normal. So this again, so both of these enzymes, these pathways for making PE are essential for life. And in each of these cases, the other pathway was performing normally. So it seems that, that these pathways for making PE provide pools of PE which have independent functions. We were unable to do many experiments in our PSD knockout mice because they died during embryogenesis, but we were able to isolate some embryonic fibroblasts. And what we found, so this is looking at mitochondria using MitoTracker Red, and you can see these are normal looking mitochondria, sort of tubular mitochondria as you would expect. However, in the knockout fibroblasts, the mitochondria were very aberrant, punctate, they were fragmented. So this would indicate that there could be some serious misfunctioning of the mitochondria in the knockout mitochondria, in the knockout cells. So obviously we were unable to do a lot of experiments because the knockout of this gene in mice was lethal, but and yet the 50% knockout, the heterozygous mice, these appeared normal. So what we decided to do was to reduce PSD mRNA by a significant amount using siRNA. And we were able to reduce the expression of this protein of the mRNA by 85%, which reduced the activity of this enzyme by about 65%. We were not able to reduce the amount of expression by any greater extent than this. And this extent of knockdown also impaired cell growth somewhat. So does this, of course, we had to check, first of all, that this knockdown did reduce PS incorporation into PE. And so we used CHO cells, and we looked at the incorporation of radio-labeled serine into PE in the control cells and the knockdown cells. And there's a great reduction in PSD activity in these cells. This knockdown also reduced modestly, I would say, the PE mass in the mitochondria. It's reduced by 25 to 30%, even though the synthesis of PE from ethanolamine was normal. So this is a rather modest reduction, but I think to reduce any more than this would be very serious defect in the cells. Incidentally, the mass of cardiolipin was normal in these cells, and the total amount of PE in the cell was essentially unchanged. We saw this effect in the mitochondria. So then we looked at the mitochondria using electron microscopy, and these are beautiful mitochondria, as you would normally see. This is using the control siRNA, but when we used the PSD siRNA, the mitochondria looked horrible. The cristae were all disorganized, the mitochondria were rounded, the mitochondrial matrix was decreased density, and the mitochondria were fragmented. So these mitochondria are not healthy looking at all. So from these studies, we conclude that this pathway, PS through MAM to PSD pathway, is required for normal mitochondrial activity in mammalian cells. We know that the import of PS into mitochondria and conversion to PE are required for mouse viability. We know also that the PE that's made from PS decarboxylase is not functionally equivalent to PE made from ethanolamine. I think that's an interesting finding. And that PS deficiency, that is using the siRNA, alters mitochondrial morphology. And I didn't show these data, but it also reduces mitochondrial PE, reduces oxygen consumption, reduces ATP production, and reduces supercomplex formation in mitochondria. And similar results have more recently been obtained by Katsu Funai's lab. He did a skeletal muscle specific knockout of PS decarboxylase and found severe mitochondrial abnormalities. Okay, so around this time, I put in a new grant application since we'd generated these knockout mice and had all these interesting studies. And actually, the grant was rejected, severely rejected, because the comment, really the only comment was that there are no human diseases involved in disruptions in this pathway. I really didn't like that. But that's how it was. Anyway, to continue, we wanted to know if, for example, if there were mutations, if there were any human mutations in PS synthase 1 that would lead to a human disease. So that's what PS synthase 1 does. It converts PC to PS. PS synthase 2 converts PE, primarily PE to PS. And I received a phone call shortly after I got that nice grant review. I received a phone call from a group in London, a human genetics group, who told me that they had found patients who have Lenz-Mazowski syndrome, and that these patients all had mutations in PS synthase 1 gene. So this is a rare disease. The children suffer from dwarfism. They have greatly increased bone mass, intellectual impairment, and other problems, particularly bones. And they had looked at 7 patients with Lenz-Mazowski syndrome who had 5 different mutations in the PS synthase 1 gene. And they wanted to know if we could find out what was the problem. So they isolated fibroblasts from the patients and sent them to us. And we set about to try and answer this question. Why do these mutations cause this syndrome? So we were a little intrigued because several deletions in the human PS, large deletions in the human PS synthase gene do not cause LMS disease. And also the PS synthase 1 knockout mice who don't have PS synthase 1, they appeared normal. So we had to decide, first of all, if the PS synthase 1 activity was reduced in these fibroblasts from the patients. And so the answer was, surprisingly, no, it was not decreased. In fact, it was increased in these fibroblasts. So this is a study where we just measured PS synthase activity by looking at the conversion of PS to PE in control fibroblasts and the Lenz-Mazowski patient fibroblasts. So you can see that the incorporation of radiolabel into phosphatidylserine. And you can see that in the patient fibroblasts, the incorporation of serine into PS is greater than in the control. It's not less, as we had predicted. So why is this? Why is PS synthesis increased in these fibroblasts? Is it because there's more PS1 protein? And this is an immunoblot, and it shows that there's not more protein, PS synthase protein, in the patients versus the controls. This is a loading control. Also, the PS synthase 1 messenger RNA was not increased, and PS synthase 2 messenger RNA and activity were not increased. So what's going on here? Why is the synthesis of PS increased? And then we remember these beautiful studies from Nishijima's lab in Japan, in which they had shown that normally the activity of PS synthase 1 is inhibited by the end product, phosphatidylserine. So we then hypothesized that in these patients, PS synthesis is increased because the end product inhibition of the enzyme by PS is defective. So I'm showing you now high resolution structure of the PS synthase 1 protein, which is a transmembrane protein. And we're looking at the mutations in the LMS patients that were discovered. So this is where they are, all in this region of the protein. Then we compared the mutations that are known from Nishijima's work to impair the end product inhibition of this enzyme. And lo and behold, these are in the similar region of the protein. So it supported our idea that perhaps the end product inhibition was impaired in these patients. So we did this experiment directly. So here we're doing an in vitro enzymatic activity of PS synthase activity. So this is in control fibroblasts. So this is in the white, the activity of the enzyme. When we add PS to the fibroblasts, this activity has almost gone. Strong inhibition of activity by the end product. However, when we did this in the LMS fibroblasts, even the basal activity is higher than it is in the control. And yet when we add the PS synthase, the PS to the outside of the cells, this activity of PS synthase 1 is not inhibited. It's a huge difference. So these data then said to us that the problem with these fibroblasts from the LMS patients was that the end product inhibition of PS synthase 1 was defective. So they call, this is sort of like a gain of function mutation. But why does this cause the disease? I have to tell you, we really don't know the answer to this question. The phospholipid content of the fibroblasts, of the LMS fibroblasts, was essentially the same as that of the control fibroblasts. So there was not an excess of PS or a decrease of PS. It seemed that they were very similar. So what does this mean? Of course, we just looked in fibroblasts, and the tissues that are most affected are the brain and particularly the bone. So we know that PS is very important for bone development and for brain development. So maybe there's a problem here, but as far as I know, this has not yet been established, unfortunately. All right, so what about the PS decarboxylase? Are there any human mutations of PS decarboxylase gene? And it appears that there are. So a colleague at the University of Calgary, Tim Schutt, he contacted me and said he runs a metabolic disease, a mitochondrial disease clinic at the University of Calgary. And he had found two sisters who have mitochondrial disease, they have mitochondrial disease, they have short stature, muscle weakness, ataxia, and other problems. And they looked at the genes that were defective, and they found that there were recessive mutations in the PSD gene, PISD gene, and that these were compound heterozygotes. And he felt that this was something to do with the deficiency. This disease was due to a deficiency of PS decarboxylase. So they isolated the fibroblasts and sent them to us. And this is the first experiment where we're using the fibroblasts with radiolabeled serine to look at the conversion of PS to PE. So this is in the control patients, and this is in the PSD patients. So there is a defect in the activity of PS decarboxylase in these patients. I'm not showing you all the data here, but the mitochondrial respiration was significantly impaired in these fibroblasts. The mitochondria were severely fragmented, and I just put here, it was rescued by the addition of lyso-PE. Lyso-PE, if you add it to the outside of cells, it seems to be directed, channeled to mitochondria specifically, and when it gets there, it's converted to PE. So this would be a way of increasing the PE, and if this was added to the fibroblasts, if the lyso-PE was added to the fibroblasts, it rescued this fragmentation. And these studies have now been confirmed in other reports of mutations in this gene. So there was a lot more work to be done here. So there are human diseases with defects in this pathway. Mutations in PS synthase 1 cause Lenz-Majewski syndrome, and mutations in PS decarboxylase also cause a human disease. In fact, this topic now of human diseases related to deficiencies in this pathway, there are becoming lots of reports of these examples. So we've shown that in LMS, this part of the pathway, conversion of serine to PS5 PS synthase, this is defective. So these are some other diseases in which defects in MAM, etc., have been reported. So CMT, that Charcot-Marie Tooth disease, this is caused by mutations in the mitofusin 2 gene. Mitofusin 2 is present very abundantly in the MAM, and it is a phosphatidylserine binding protein. So it's thought that this disease is due to a defect in this process. In other human diseases, several neurodegenerative diseases also becoming apparent that there are defects in forming the MAM, either too much MAM or too little MAM. For example, Alzheimer's disease, Parkinson's disease, also type 2 diabetes and cancer. There's some indications. So obviously a lot more needs to be done here. And then finally, there's this mitochondrial disease caused by mutations in PS decarboxylase. So as I mentioned at first, when we discovered MAM, there was very little interest in the topic, but now this has expanded into a huge field, many labs working on membrane contact sites. So I just want to summarize a bit of the what's happening in the field. There are now contacts that have been observed between other membranes such as ER, we know ER and mitochondria, ER plasma membrane, et cetera, endosomes, lysosomes, lipid droplets. And these are becoming of great interest to metabolism. So these contacts are thought to be formed by formation of transient reversible protein tethers. So between 10 and 80 nanometers distance between the two organelles. These protein tethers, they're starting to be analyzed, what components make up these tethers. And it's a large field, and I haven't spent any time talking about it. I just want to say that there seem to be some similarities between the tethers of the different types of membrane contact sites. For example, structural proteins that bridge two membranes seem to be a frequent component. Functional proteins such as ion channels, lipid transfer proteins seem to be components of these tethers. An interesting finding is that the inositol trisphosphate receptors that mediate mitochondrial uptake of calcium from the ER into mitochondria, right? These are highly enriched in the MAM. So this is thought to occur in the MAM, this process. And of course, we are particularly interested in lipid trafficking. It seems that there are other examples of interorganelle lipid trafficking that could be using these contact sites. And for what about cholesterol, for example? So I want to just highlight two recent papers that indicate a strongly indicate the involvement of ER plasma membrane contact sites in the movement of cholesterol from the plasma membrane to the ER. So the first one of these was from Peter Tontono's lab, and he discovered these proteins, the aster proteins. So these are proteins that are in the plasma membrane, and they bind cholesterol and phosphatidylserine. So when HDL, high density lipoprotein, delivers its cholesterol to the plasma membrane, these aster proteins cluster in the plasma membrane and cause the formation of a contact site with the ER, thereby delivering the cholesterol from the plasma membrane to the ER. So this is an interesting finding, and I think it's important. The other study was one that came out from the Brown and Goldstein lab about a month or two ago, and it's concerning the delivery of cholesterol from low density lipoproteins. So LDL bind to the cell surface at the LDL receptor. The lipoprotein is internalized, delivered to the lysosomes. From there, the cholesterol from the LDL is delivered to the plasma membrane. It's eventually delivered to the endoplasmic reticulum via an excursion through the plasma membrane. Most of the cholesterol, in fact, that ends up in the ER goes through the plasma membrane. And what they have shown is that this process occurs readily if phosphatidylserine is abundant in the cells. However, if PS synthase 1 is depleted and the PS content of the cells is reduced, the cholesterol, once it's in the ER, once it's in the plasma membrane, is not delivered to the ER and accumulates in the plasma membrane. So this occurs when they knock down phosphatidylserine synthase 1, but not PS synthase 2. Interesting finding. So it seems that PS is regulating the transfer of cholesterol from the plasma membrane to the ER. So future directions, of course, we need to have a lot more studies on the lipidomics and proteomics of these contact sites and the tethers. We need to know, especially need to know how contact site formation is regulated. It appears to be regulated partly by metabolic state of the animal or the cell. There are some indications that anionic lipids such as phosphatidylserine and phosphatidylinositol phosphates promote the formation of membrane contact sites. Is this a key finding? And also, of course, which membrane contact sites mediate intra-organelle lipid transport? Which lipids, and is this a bidirectional process? So those are the questions for you guys to start working on. And I'd like to thank the people who did this work from my lab, these various students and postdoctoral fellows and technicians, wonderful workers, and also these collaborating labs. Thank you.
Thank you, Jean. That was fantastic. It's great to hear how much is known, but also how much isn't known, and really inspiring for people working in the field now to think about future directions to go in. I think there's so much also that isn't known about how all these things respond to acute challenges for, you know, increasing mitochondrial number and mitochondrial function, and how all the lipid biosynthetic pathways respond to that. There must be a lot of work still to be done on that, I guess. Yeah, how these contact sites.
You're absolutely right.
Yeah.
Can you hear me?
Yes.
Okay.
Yeah,
you're absolutely right.
Yeah.
There's a lot to be done. I mean, there is.
So we've got loads of questions. So I'm going to start with Ali Hajaya, who asked 2 questions quite early on. So Ali, you should be able to talk now.
Hello.
Hello.
Thank you for the talk. It's really exciting because I just got in the field of phosphatidylserine.
Okay,
I
good.
have a couple of questions. So the first thing is a quick one. Does PEN methyltransferase also accept PS as a substrate?
No, not that I know of. And Dennis is actually sitting next to me, and he is the expert on PE methyltransferase, and he says no.
One other question, if you don't mind. So you showed that in the knockout of phosphatidylserine, decarboxylase, there is mitochondrial fragmentation. Have you done any measurements of membrane charge, for example, by fluorescence? Because it could be due to PS being continuously imported, like being imported into mitochondria, and then the charge concentrate, and then there's an imbalance. I don't know if you've done.
This is in the knockdown cells, right? The siRNA cells. I don't think that the PS accumulated that much in those cells.
Oh, okay.
I have to go back and look at that. I'm sorry, I can't remember.
Okay, so thanks for
Thank you.
Pavitra. So I think I unmuted Pavitra. Yeah, okay, go ahead. Ask
Hello,
your
are you
question.
able to? Thank you very much. Thank you very much, Jane. And my question is, does the mitochondria membrane potential affects area of MEMS? For example, the contact area, which is mediated, which is between ER and mitochondria.
Is it what? Sorry, I couldn't hear.
Does mitochondrial membrane potential affects the area of MEMS?
Oh, good question. And I'm not sure of the answer. Actually, that's a good question. Sorry.
Thank you.
Okay, so I'm now looking for Stephen Fleisler. I'm just unmuting you.
Can you hear me?
Yeah.
Excellent. Hi, Jean. Greetings from Buffalo, New York.
Hello.
Loved your talk. I had two questions. The first one was, what is the specific step that requires ATP to
Yeah. So actually, Dennis Volker has done quite a bit of work on that. We didn't really investigate too much, but it seems to be, I mean, they don't really know, but it seems to be just the fact of bringing the two membranes close together. That's what is the theory.
So that I had asked
ATP is required for that. Yeah.
Yeah, I'd ask if maybe a flipase might be involved.
I see what you're saying. Yes, yes, that's a good idea. It could be, I mean, so that they'll only come to, yes, and actually there were some original studies from Ardai's work in France showing that these ER mitochondria contact sites occurred at places where the inner and outer mitochondrial membranes came together. So I don't know if that's relevant either.
Okay.
And if that really is the case, if that's what a common occurrence, I don't really know. So that's a good idea. I don't know about that. I think that nobody's really investigated that too much detail. Dennis Volker would be the person who would know the most.
Okay. And the second question was, in the subcellular fractionation experiment you described, so you're looking for an enzyme that is enriched in MAM, but it's very low in ER and it's low in mitochondria. So how do you explain that enrichment? Is there a retention signal or targeting or what?
Well, you know, the ER is not a homogeneous distribution, does not have a homogeneous distribution of proteins. You know, there are other regions of the ER that are specialized in certain proteins. And of course, if you analyze microsomes contain MAM, right?
Yes.
But purified ER fractions, like rough ER and smooth ER, they contain different distributions of proteins. And MAM is another area of the ER that's enriched in certain proteins. And I mean, who knows why? It's a good question. Is that what you wanted me to say?
Sort of. Maybe I could wrap my head around it even more if I could understand. Has anybody done a calculation for what percentage of the total ER-derived membranes is accounted for in MAM? It seems like it would be pretty low percentage.
Yes, I think that could be. I think we don't know the answer to that. I think it is pretty low. I mean, it's not 50% or anything like that. No, it's much lower than that.
Got it.
Got loads of questions. So yeah, thank you, Stephen. Stephen Sturley.
Oh, Steve Sturley. Hello.
Yeah.
Hello, Steve.
Hi, Jean.
Yes, hello, Steve. Good to hear from you.
Wonderful, wonderful talk as usual. Good to see you and Dennis. I had a small question, which was to ask if the gain of function mutations that you showed in the human syndrome are dominant or recessive.
In which, sorry, I'm sorry, say that again.
In the human syndrome, I forget the name of it, where you showed that you had a gain of function mutation, where you got elevated levels of...
Lenz-Majewski syndrome.
Yes, right.
Recessive.
So does that fit genetically with what you would expect for a gain of function mutations? I would have thought they might have been dominant.
Oh.
Maybe suggesting there's a complex involved, or...
Yeah, I mean, I think I'm right on that. Actually, no, I think they are dominant. I'm sorry, I was getting confused by the PSD carboxylase. Yes, I think they're dominant in Lenz-Majewski. All
right, good, that makes sense.
And I think they're also, yes, that's exactly right. I'm sorry, I made a mistake there. Yep.
Okay, that makes sense, good. I had just a general question to follow up on the energy transfer, energy requirement. So you showed nicely that you need energy, as the previous questioner asked. What happens to all the other membrane contact sites in those energy poison cells? Do they disappear or get malfunctioning as well?
Well, you know, so little is known about these other membrane contact sites, and I think that that's something that needs to be done. You know, like the ER plasma membrane, right? For example, those are fairly well characterized in a way, but I don't think anything's known about the energy requirement for that.
Okay. Right. Yeah, thank you.
Okay, thank you very much. Shamshad Cockcroft.
Oh. Hello, Shamshad. Hello. You there?
I think he just left.
Okay.
Just as I was. Oh no, here he is.
Okay, sorry, I had to unmute myself. Hello.
Okay. I thought I had done that. Sorry, apologies. It's difficult on this panelist list because as soon as you unmute people, they jump to a different part of the list and then I can't find them anymore. So you're back now. Good. So go ahead.
Hello.
So my understanding of it is the MAM is part of the is the ER component.
Correct.
A specialized ER component. So you still need to get the PS across to the mitochondria. So what is the mechanism for getting the transfer of PS to the mitochondria?
Yeah, yes, well, I mean, you have to have this contact site. So it is, I think that the idea is that these tethers bring the two membranes together. It's not fused, but they bring the membranes close enough together that the lipid can get to the other membrane without having a huge energy barrier, you know, preventing it.
Lipids are not going to jump out of a membrane.
Oh, well, probably there are, in many of these tethers, there are lipid transfer proteins that make up part of the tether. And so I think the general idea is that some of these lipid transfer proteins are involved in doing this. It's a sort of a wishy-washy answer, but I think that's...
There is a protein that's been identified called VAT1, which is a PS soluble PS transfer protein.
Yes.
Do you think that that is involved in the PS transfer?
Could well be. It could well be. I haven't seen what happens when you delete this protein.
Okay.
Okay, thank you.
That would be a good experiment. Delete that and see what happens.
Okay, so I think we probably have time for another two because we're running out of time. So Grant Hatch.
Ah, okay. Hello, Grant.
Hi, Jean. Great talk.
Hello.
Hello from Winnipeg.
Yes.
So, and say hello to Dennis for me, please. So this Lenz-Majewski syndrome,
Yes.
do you think in terms of a mechanism for how PSS is inhibited, do you think it's the PS electrostatic interaction is disrupted on the PSS protein? That the mutation that occurs, does the phospholipid itself, its charge, is it involved in maybe inhibiting the PSS, so it's not able to interact properly with the PSS molecule?
Yes, I think something like that. These studies were done, quite extensive studies done by Nishijima's lab and the various people whose names I put on the slide. But I think they didn't really come to a firm conclusion about that.
Okay.
Yeah, but I think that is, you know, the protein is going to be somehow folded differently. So,
Right. Mhm.
Yes, and so
Okay.
that the PS perhaps can't get to this site and bind to where it should do or whatever, right? Yeah, that's what I think is happening, but I'm not sure.
Okay, thank
Okay,
you.
thanks, Jean.
So one last question from Kevin Titeka.
You can skip to someone else because my question was already answered. It was about the ATP use.
Okay. Okay.
Maybe you have an answer. Do you have an answer?
I don't have an answer, but I do know that LMS is autosomal dominant.
Yes, I did say that in the end.
Yeah. Okay.
Yeah.
Okay. So one last question from Sriganesh Rao.
Oh yes. Hello.
Takes a second for people to come on when they're unmuted.
Hi. So just a simple question. How does this protein tether mechanism differ from a snare-mediated vesicle organelle fusion, which would simply allow mass mixing of membranes? How is it that there is a selectivity to what lipids are transferred from ER to mitochondria?
So I'm not exactly sure I understand, but it isn't a fusion between the two membranes. It's not a fusion. Because you could separate the MAM from the mitochondria, yes, from the mitochondria, just doing centrifugation, you know, on Percol or whatever, or by treating with high salt or something like that. So it isn't a fusion. Okay.
Thank you. Thank you for that.
One idea might be that people have had is that there could be a protein in the MAM that prevents membrane fusion, or in the ER, or you know, or somewhere that prevents the membrane fusion occurring. I don't know if that's a possibility, but that has been suggested.
Thanks.
Okay. Okay, thanks. So, well, we're out of time now, Jean. So we'll need to wrap up. But I just wanted to mention that Dennis has posted that the 6th edition of the textbook was published in 2015. I apologize, I got that wrong. So there's a more up-to-date version. And when I went on Google, maybe you switched publishers, because I don't think it came up on the site that I looked at, which was.
It's Elsevier. It is Elsevier. But Dennis and I are no longer the editors.
Ah. Okay, edited by Neil Ridgway. So that's the 6th edition, and that's why I didn't find it. So yeah, so that's the follow-on. Okay, thanks for clarifying, Dennis.
Go out and buy it.
Yeah, well, we buy the one that you wrote. Anyway, thank you for a fantastic talk. There's been so many questions. I'm sure people will be emailing you afterwards with the questions, because there was a lot that we didn't get to answer. It's always a sign of a really good talk. And I think it's really, this is a great field because there's so much to do. It's so interesting, and there's so much to do. Yeah.
So much to do.
Absolutely. Yeah. So it's been a real pleasure to host you today, Jean. So thank you very much, everybody. Yeah. Goodbye.
Bye.